Now that we have a basic understanding of what happens during cellular respiration via the reactions of glycolysis, pyruvate oxidation, the citric acid cycle, and oxidative phosphorylation, I'd like to spend some time focusing on the regulation of this central metabolic pathway. As we've discussed before, intermediary metabolism describes the situation that we see in cells in which catabolic, or breaking down reactions, or pathways, are linked to anabolic, or building up reactions, or pathways, via intermediates that are common to both. In this diagram, we see intermediates linking the catabolism and anabolism of the four classes of biomolecules that make up living cells: proteins, nucleic acids, carbohydrates, and lipids. Running down the middle of this diagram is cellular respiration. Here's our starting sugar glucose being oxidized to 2-pyruvate through the reactions of glycolysis. Pyruvate oxidation forms CO2 and acetyl, which gets linked to coenzyme A, and the acetyl then gets oxidized through the reactions of the citric acid cycle, forming CO2. Along the way, we know that linked to the oxidation of glucose and glycolysis, we see reduction of NAD+ to NADH and the formation of 2 ATP net. More NADH is produced in pyruvate oxidation, and in the citric acid cycle, both NADH and FADH2 are produced, along with another 2 ATP equivalents for every glucose that we started with. At the end of this long metabolic pathway, we see the energy being carried by these coenzymes, NADH and FADH2, being harvested through oxidative phosphorylation and used to synthesize the bulk of cellular ATP. But what this diagram is illustrating for us is that cells can not only use sugars for this metabolism, they can also use lipids and proteins and even nucleotides. If we take a look at lipids over here on the right, we know that fats are made up of a glycerol molecule linked to 3 fatty acid molecules forming a triacylglyceride. The glycerol and fatty acids can be hydrolyzed enzymatically, and the glycerol can then be converted by an enzyme to form glyceraldehyde 3-phosphate, which we know is an intermediate of the glycolytic pathway, and so it can then proceed through the rest of glycolysis. The fatty acids, which have long hydrocarbon tails, consisting usually of 16 or 18 carbons, get enzymatically cleaved into 2-carbon acetyl units, which can then feed into the citric acid cycle. Likewise, taking a look at proteins over here on the left, proteins get broken down into individual amino acid monomers, which can then be hydrolyzed to deaminate or remove the amino group, leaving behind what is essentially a carbon skeleton of varying structure depending on which amino acid we started with. These carbon skeletons can be enzymatically converted to various intermediates of cellular respiration, with some of them being converted to pyruvate, some being cleaved to form 2-carbon acetyl fragments, and some being enzymatically converted to various intermediates of the citric acid cycle. But notice, the arrows go in both directions. So if energy is abundant in the cell, then the cell can take these intermediates of cellular respiration and use them to synthesize amino acids, to synthesize glycerol and fatty acids, and therefore to participate in anabolism. So depending on the changing needs of the cell, metabolism shifts back and forth via the regulation of enzymes to favor oxidation and ATP production or biosynthesis. Now let's take a closer look at what that regulation looks like in glycolysis. Based on what you've learned so far about feedback inhibition, pause the video and take a moment to consider how that might work in glycolysis. Remember that feedback, or end product inhibition, is when an enzyme that functions in a pathway is inhibited by a product of the pathway, thereby slowing the pathway down when there's plenty of product. So what are the products of glycolysis that could hypothetically act as inhibitors at an earlier step in the pathway? Well, we know that pyruvate is a product, NADH is a product, and ATP is a product. Any of these would make sense as an inhibitor. Well, what enzyme makes sense as a point of regulation? Typically, key rate-limiting or energy-intensive steps are the main points of regulation. So if we take a look at the reactions of glycolysis, we might identify the first energy investment step catalyzed by hexokinase, or the third reaction catalyzed by phosphofructokinase as likely points of regulation. But if we think a bit deeper, we can see that it makes more sense for the cell to regulate PFK than hexokinase. Remember that the phosphorylation of glucose by hexokinase allows a cell to trap glucose in the cell, and then, based on energy needs, the cell can either store it or oxidize it to make ATP. If hexokinase were inhibited, the cell would lose that advantage. PFK, however, catalyzes the commitment step for glycolysis. Once this sugar gets phosphorylated a second time, it will proceed through the rest of the pathway. So it makes much more sense to regulate that rather than hexokinase. And that is, in fact, the main point of enzyme regulation in the glycolytic pathway. And as it turns out, ATP is a critical regulator of PFK function. When ATP concentrations rise in the cell, ATP acts as an inhibitor of phosphofructokinase, which slows its activity, and not as much fructose 6-phosphate is phosphorylated to produce fructose 1,6-bisphosphate. Other regulators also influence PFK. Citrate, which is an intermediate of the citric acid cycle, also inhibits PFK, and adenosine monophosphate, or AMP, acts as an activator of the enzyme. This makes sense because if citrate levels rise, essentially that's an indication that the citric acid cycle isn't keeping up with glycolysis. So it makes sense that citrate would act to slow down glycolysis. AMP levels rise when ATP levels fall in the cell. So this signals the pathway that energy reserves are low, and the cell needs more glycolysis taking place to regenerate the pool of ATP from AMP and ADP. But the inhibition of PFK by ATP actually presents us with a bit of a puzzle. Remember that PFK catalyzes the phosphorylation of fructose 6-phosphate using ATP. Essentially, if we imagine this is the PFK enzyme, and here's the active site, within the active site there's a binding site for fructose 6-phosphate and another binding site for ATP. And the enzyme catalyzes the transfer of phosphate from ATP to the sugar, resulting in the products fructose 1,6-bisphosphate and ADP, which are then released by the enzyme. So, if ATP is a substrate of the enzyme, how can it also act as an inhibitor? Could ATP function as a competitive inhibitor in this case? Or does it make more sense that ATP would function as a non-competitive inhibitor? How does ATP only inhibit the enzyme when it's at high concentrations, but not low concentrations? It's difficult to imagine how ATP could function as a competitive inhibitor. If it enters the active site and fructose 6-phosphate is also present, the ATP will be acted upon as a substrate. Remember that competitive inhibitors act by blocking access of the substrate to the active site. Hypothetically, ATP could act to block fructose 6-phosphate entry to the active site, which would have the effect of inhibiting the reaction, so in that case it could make sense for ATP to be a competitive inhibitor. But as it turns out, ATP does not bind to the fructose 6-phosphate binding site in the active site, so it does not competitively inhibit that way either. Instead, what we see is a second binding site for ATP on the enzyme, an allosteric site away from the active site. When ATP binds here, the properties of the enzyme change so that catabolism slows down and we see inhibition. But how do we account for the fact that ATP only inhibits at high concentrations? And conversely, when ATP is present at low concentrations, it is acted upon as a substrate. To understand this, we need to envision how binding occurs. Remember that there has to be an attraction or affinity between the interacting molecules. Complementary hydrogen bonding functional groups, or groups that can form ionic bonds, or even hydrophobic functional groups that get pushed together due to water repulsion. So this ATP in the binding site is binding due to the interactions occurring between ATP and the amino acid R groups in this region of the protein in the active site. Likewise, the same is occurring here at the distal allosteric site. So if the interaction involves a strong attraction or affinity, then binding is going to happen at lower concentrations, because if ATP gets anywhere near the interacting functional groups on the enzyme, we'll see that attraction taking place. On the other hand, if the attraction between the enzyme and ATP is relatively weak, then binding is less likely to occur unless there's a lot of ATP around, making it more likely for that ATP to contact the enzyme in just the right orientation to drive binding. So given that understanding, and knowing that ATP is acted upon as a substrate at lower ATP concentrations, and only binds to the allosteric site and inhibits the enzyme at higher concentrations, we can hypothesize that the ATP binding site within the active site is a high affinity site, and the distal site is a low affinity site, and that turns out to be exactly correct. So this explains why ATP only binds the inhibitory site when concentrations of ATP go up. For full disclosure, there are other points of regulation in glycolysis and other regulators besides ATP. You'll learn more about them if and when you take your upper division biochemistry courses. Now let's turn our attention to regulation of the citric acid cycle. Using the same logic we just went through for glycolysis, hypothetically it's possible for nearly all of the products of the citric acid cycle to act as feedback inhibitors, with the exception of carbon dioxide. So NADH, FADH2, GTP, or ATP could act as feedback inhibitors. Why not carbon dioxide? Well, remember that CO2 is a non-polar molecule. It freely diffuses across phospholipid bilayers, so it doesn't accumulate. It turns out that for the citric acid cycle, NADH is a key inhibitor of several enzymes. These red stop signs represent enzymes that are inhibited by high concentrations of NADH. So we see inhibition of multiple citric acid cycle enzymes, and we also see inhibition of the enzyme that oxidizes pyruvate to acetyl, pyruvate dehydrogenase. There are other regulators as well. ADP and calcium both activate reactions of the citric acid cycle and PDH, but NADH regulation is key. So what effect would this inhibition have on the metabolism of the cell? Remember that diagram illustrating intermediary metabolism that we saw at the start of the video, and those arrows that go both ways towards catabolism and towards anabolism? When NADH levels go up and the citric acid cycle slows, intermediates of the cycle get diverted to those other biosynthetic pathways. So we can see that intermediates of the cycle, like citrate, can be siphoned off and used to synthesize fatty acids and cholesterol. Alpha-ketoglutarate can be used to synthesize some amino acids. Likewise for oxaloacetate, and so on. So again, if the energy needs of the cell are being met, there's no advantage to continuing to oxidize biomolecules to produce more ATP. But there is an advantage to shifting the metabolism of the cell to storage and synthesis, and that's exactly what we see taking place. This allows cells to respond to dynamic and constantly changing conditions in the cell, and to effectively maintain homeostasis and energy balance. If we zoom back out and think about this regulation more holistically, this also explains the shift in metabolism that we see happening when oxygen becomes limiting in the cell, which we know leads to fermentation and the use of glycolysis as the main source of ATP. Remember that oxygen acts as the final electron acceptor in oxidative phosphorylation. It receives electrons from complex 4 of the electron transport chain, picks up protons, and forms water. These electrons originated from the reduced coenzymes NADH and FADH2, and the movement of electrons down the electron transport chain is directly responsible for the movement of protons from the mitochondrial matrix into the intermembrane space, building a proton concentration gradient. This gradient and the movement of protons down their gradient through ATP synthase forces the shape change in ATP synthase that powers the formation of ATP from ADP and phosphate. If oxygen is limiting, and complex 4 has no recipient for the electrons coming from further up in the chain, then electron transport can't happen. If electron transport isn't taking place, then protons aren't being moved to the intermembrane space, and the concentration gradient that's used to power ATP synthesis isn't being established, so ATP won't be made. So oxidative phosphorylation no longer takes place in the absence of oxygen. But realize also that what this means is that NADH and FADH2 won't be oxidized, so concentrations of NADH will rise. As we just learned, the consequence of that is that the citric acid cycle is now inhibited as well. So the small amount of ATP equivalents produced during the citric acid cycle are also not going to be produced. The only source of ATP synthesis in the cell becomes glycolysis. The other thing to remember is that NADH also inhibits pyruvate dehydrogenase. So pyruvate won't be oxidized in the mitochondrion, and pyruvate concentrations in the matrix will rise, which slows the transport of pyruvate from the cytoplasm into the mitochondrion. So we'll see a buildup of pyruvate in the cytoplasm of the cell. This also helps trigger the metabolic switch from respiration to fermentation, because it turns out that the enzymes that catalyze fermentation are activated by rising concentrations of their substrate, pyruvate. Remember that the function of fermentation is to regenerate NAD+ so that glycolysis can continue to occur. Depending on the organism, either lactic acid fermentation or ethanol fermentation is going to take place. In organisms like yeast, we see ethanol fermentation taking place. First, pyruvate is oxidized to form acetaldehyde, which also yields CO2. This acetaldehyde is then reduced to form ethanol, and this reduction is linked to the oxidation of NADH back to NAD+, providing the coenzyme needed for glycolysis to continue. In organisms like animals and some bacteria, lactic acid fermentation takes place instead, and we see pyruvate being reduced to form lactate. And again, this reduction is linked to the oxidation of NADH back to NAD+, providing the essential coenzyme required for glycolysis to continue. So under conditions when mitochondrial ATP production isn't meeting the needs of the cell because oxygen is limiting or energy demands are just too high, the cell's metabolism shifts to fermentation, which produces less ATP per glucose, but produces it much more quickly. It turns out that these enzymes that catalyze the conversion of pyruvate to lactate or acetaldehyde are positively regulated by pyruvate concentrations. That is, as pyruvate concentrations increase in the cytoplasm due to the backup occurring in the mitochondrion, this causes an increase in these fermentation reactions, which increases the amount of ATP being synthesized via glycolysis. So not only do we see feedback regulation occurring within the glycolytic pathway and within the mitochondrion, but also more globally between the mitochondrial reactions and the reactions happening out here in the cytoplasm. In this way, the cell can be responsive to changing cellular conditions and concentrations of ATP, NADH, and various metabolites, which leads to greater efficiency in maintaining homeostasis and meeting the energy demands of the cell. Again, for full disclosure, we just scratched the surface on this regulation. It gets much more complicated, but this discussion should give you a basic understanding of how it works, and those concepts hold when you dig deeper into the details of metabolic regulation.